type the topics which I covered in the first unit. This is the title of the first unit, Introduction to Drug Design and Discovery. The first topic is drug discovery, therapeutic targets, identification and validation. Second topic is drug developmental process. Outline. And the third topic is discovery of drug candidates. So the fourth topic is sources of hits, leads, and candidate drugs. The next topic is basic principles of lead development and optimization. Next topic is membrane penetration and Lipinski's rule of 5. Next topic is stereochemistry. And their importance. Okay, so these are the topics given in the syllabus in the first unit, right? So I hope all the topics very extensively we have covered, right? Can you check and tell whether we missed any topic in the first unit? Target identification, validation, we covered. Various stages of drug discovery process, we covered. How to discover the drug candidate, I explained. Sources of hits, leads, candidate drug, covered. Basic principles of lead development and optimization, covered. Membrane penetration, Lipinski's rule, I covered. Stereochemistry and drug design, I covered, right?
Yes, sir.
Okay, I think all the topics covered. All the video lectures already available. You can go through that. Unit 2, we are moving to Unit 2. The title of Unit 2 is Docking, Pharmacophore, and QSR models. Okay, so this unit is very important, right? So because so these units very extensively cover the various aspects of computer aided drug design. Okay, so very important unit, right? So many practical aspects will be covered in this unit related to the computer aided drug design, right? So before the first topic of this unit is role of X-ray crystallography in drug design, structure guided drug design. Role of X-ray crystallography in guided drug design. Okay, this is the first topic, right? So which I am going to talk today, right? So before going for before saying the role of X-ray crystallography in drug design, right? So I just wanted to give a very brief introduction about what is meant by computer aided drug design, because all this docking, pharmacophore, KSR model, all this comes under one umbrella called as computer aided drug design. Okay. So computer aided drug design. I hope a few of you attended the workshop conducted by Schrodinger and Sastra, right? So the primary talk by different people in that workshop, I hope that is from computer aided drug design called as CAD. Okay, so why CAD? What is CAD? So we know that drug discovery process is a very time consuming process. Also, a lot of money has to be invested, right? So in some way, if you can accelerate that, reducing the money and reducing the time taken for discovering the drug molecule, that is actually a good thing, right? So in that aspect, we can say that computers plays a very important role in drug discovery process. So wherever you use the computer for accelerating the drug discovery process, for reducing the money or for reducing the time, then all those comes under one concept called as computer aided drug design, CAD, right? There are variety of applications in CAD which can be very well utilized during the drug discovery process, starting from understanding the disease, identification of the target, validation of the target, identification of the hit molecule, of lead optimization, and even ADMET prediction, simulation, so many things there. All stage almost, right? So except the clinical trial, all stage of the drug discovery process, we can use this computer aided drug design. And also we can do some kind of simulations related to clinical trial. Okay, that is also possible, right? So which means that if you look at the pipeline of drug discovery process, almost you are free to use anywhere the computer to accelerating the process. If you do so, that is what is called as the computer aided drug design, right? So basically, we can classify this computer aided drug designing into two major classes. One is called as structure based drug design. Hope the same is covered in the Schrodinger workshop also. Structure based drug design. So another aspect is called as ligand based drug design. Ligand based drug design. So hope my screen is clearly visible, right? What I'm writing.
Yes, sir.
Yeah, okay. So there's another aspect called as combination of this structure based drug designing and ligand based drug designing. They call it as amalgam of, I write it short as structure based drug design, SBDD, and ligand based drug designing as LBDD. Combination of both structure based and ligand based, right? So anyway, these are the two broad classifications: structure based drug designing and ligand based drug design, right? So let's see what is meant by structure based drug design and how does it differ from ligand based drug design. Let's start with structure based drug design. So I'll first give example for structure based drug design, so it will be easy for you to understand what exactly is the meaning of structure based drug design, right? So if we take docking, molecular docking, we already started learning molecular docking in the laboratory session. Flexible docking, rigid docking, all these are example for structure based drug design. And if we say that protein modeling, which comes under part of structure based drug design. Okay, so example for ligand based drug design, so we can say the pharmacophore given in the title is an example for ligand based drug design, or QSAR is an example for ligand based drug design. So what's the example for amalgam of structure based and ligand based drug design? If you take de novo ligand design, sometimes we can call this as structure based drug design, but normally it uses both the concept of structure based drug designing and ligand based drug design. Okay, these are some examples of structure based drug design, ligand based drug design, and de novo amalgam of structure based and ligand based drug design, right? So coming to structure based drug designing, how does it differ from the ligand based drug design? It is very simple to remember what is meant by structure based drug design. Okay, the general concept, whatever we have studied so far. So we know what is meant by drug molecule. We know what is the meaning of receptor, and we know that all the drug molecules in modern day, right? So we explained the concept as the drug molecule should go and bind to the receptor to form drug receptor complex. Okay, so the formation of this complex lead for the biological response. So this is our understanding so far, right? So what actually happens is that the drug molecule should go and bind to some receptor to produce the response. So if we take aspirin, aspirin molecule should go and bind to the receptor. Uh, you know what is the receptor? Corresponding receptor for aspirin. Somebody can mute and somebody can unmute and tell. What's the drug target for aspirin? So I think we discussed about drug action of aspirin, right?
Yes, sir.
What is the molecular target of aspirin? Okay, the molecular target of aspirin is cyclooxygenase. Okay, so it forms aspirin cyclooxygenase complex. So that lead for the biological response. The biological response of aspirin is it relieves the pain. It reduces the pain, inflammation, the many activities for aspirin. So what is going to be structure-based drug design? If you design a drug molecule based on the 3D structure of the drug target. So here the example is cyclooxygenase is the example, right? So once if you know what is the 3D structure of the cyclooxygenase, say this is the 3D structure of the cyclooxygenase, and once if you design the drug molecule based on the 3D structure of cyclooxygenase or based on the structure of the receptor or the protein, then that approach is what is called as the structure based drug design. So we can say that if we want to proceed with structure based drug design, we should definitely know what is the 3D structure of the drug target. So without knowing the 3D structure of the drug target, we cannot proceed with the structure based drug design. It is mandatory to know what is the 3D structure of the drug target. And starting point of structure based drug designing is from the 3D structure of the drug target. So now you may have a question that if we want to know the 3D structure of drug target, okay, so how we can get to know about the 3D structure of the drug target? There are a lot of experimental methods which are available to determine the 3D structure of the drug target. So some of this method you may already studied in detail very extensively in some other coursework like X-ray crystallography. We can use nuclear magnetic resonance spectroscopy. We can use electron microscopy. Three common methods I have mentioned here. Many more methods there. So these are the methods by which we can solve the 3D structure of the drug target. We can get to know about the 3D structure using any of these methods like X-ray crystallography, NMR, electron microscopy. So solving the 3D structure of the drug target. So once if you solve the 3D structure of the drug target of your interest, then we can use those structure as the starting point for the structure-based drug design. But this is not always possible. Okay, not always possible. So why do I say it is not always possible? The reason is that there are some cases in which we may not able to determine the 3D structure of the drug target. So if we take GPCR, it's called as G protein coupled receptor, right? So it's a family of protein comes under receptor. So the main function of a receptor is that it takes the signal from extracellular part of the extracellular region and transmit that information to the intracellular region, right? So say if you think this is the lipid bilayer membrane, cell membrane. Okay, so we can differentiate two regions here. Say this is the extracellular region and this is the intracellular region. Say some proteins, they may be embedded in this membrane. So those protein molecules are called as normally the transmembrane proteins, the proteins which are embedded, right? So here this illustration is some transmembrane protein. GPCR is one such transmembrane protein. So if you look at the structure of this protein, some region is present in the extracellular region and some region is present in the intracellular region. So the signaling molecules messenger will come and bind to the extracellular region of this receptor, and that information signal carried by the messenger will be transmitted inside the cell. Signal transduction, this is what is called as signal transduction, right? Okay, so coming to why we discussed about this GPCR, right? So most of the disease in our body is caused because of some malfunctioning of GPCR. If you look at the pie chart of the disease causing or macromolecules involved in disease causing, right? So majority of the portion normally occupied by this G protein coupled receptor. And since this is a membrane embedded protein molecule, it will be very difficult for you to extract this protein molecule from the cell. There will be a strong attraction between the helices and the membrane region. Okay, so it will be very difficult to isolate this fully successfully, to go for either X-ray crystallography, NMR, or electron microscopy, because all these methods we need purified form of the protein, full length protein molecule, but it's very difficult task for doing that, right? So it's very difficult to isolate, purify, going for structural determination, right? But as I said, that GPCR plays a very important role disease, okay, which are caused because of some malfunctioning of this GPCR. Then we cannot proceed with structure based drug design. But we know very well that it's a very important protein involved in causing disease. So what is the alternate method? The alternate method is what is called as the ligand based drug design. The alternate method is ligand based drug design. So in case if we are not able to determine the structure of 3D structure of the drug target, there's alternate method in computer aided drug design called as ligand based drug design. So what do we do normally in ligand based drug design? So you know so there are two types of drug design. Already we discussed this in detail. Okay, so generally we go for one type called as can you tell what are the two types we have discussed? Let's put this as a question. Two types of drug design. In target validation identification, I discussed about two major types of drug design. What is that? Again, I repeat, during the target validation identification, I discussed about two major types of drug design. And I think I gave the material, right? So, in which also I discussed in detail about the two types. What is that? There are cases in which, you may know the drug molecule, you may not know what is the drug target. Again, I repeat, there are cases in which you may know what is the drug molecule, but you may not know what is the drug target. The classical example is aspirin. Okay, for long period of time, more than 30, 40 years, right? After marketing the drug molecule, there is no clue about what is the molecular target of aspirin, but it is marketed, approved by FDA, marketed it as a drug molecule, but without knowing what is the drug target. Again, I repeat, very important point. FDA approved the drug molecule to market. FDA knows that this molecule can reduce the pain, fever, whatever it is, inflammation, whatever it is. But there is no clue about what is the molecular target. After 30, 40, 50 years, some scientists who discovered that this is the molecular action of aspirin. That is what we have discussed in the drug action of aspirin. Right? So in that case, we know the drug molecule, we do not know what is the molecular target. Do you agree? We know the drug molecule, we do not know what is the drug target. So based on the phenotypic change, which is observed, so we know that those molecules have the ability to cure the disease. Okay, so that is the base for ligand based drug design. What we need is we need set of ligand molecule. We need set of ligand molecules with known biological activity. I just make it simple, right? So don't take uh, in deep, right? So just for understanding, say I have set of molecules. So these molecules can reduce the fever. Okay, this molecule can reduce the fever. So based on the phenotypic change, I observed that while giving this molecule, there is a reduction in the fever. They're able to reduce the fever. But I do not know what is the molecular target. Even I don't have any clue about what is the molecular target to which the molecule interact, how it is reducing the fever. We don't have any clue. But these are the set of molecules. They're capable of reducing the fever. Okay, so we can use this set of compounds for designing a new drug molecule. If we do so, that approach is what is called as ligand based drug design. So what is ligand based drug design? So we know set of molecules and we know what's the biological activity of that molecule, reducing the fever, but we do not know what is the molecular target. So we are going to use the set of molecule to design new drug molecule. If you do so, that approach is what is called as the ligand based drug design. So let's simplify this, right? So difference between structure based drug design and ligand based drug design. So in case of structure based drug design, we should know the 3D structure of the drug target. In case of ligand based drug designing, it is not necessary that we should know what is the 3D structure of the drug target. Okay, so in case of ligand based drug designing, we should know set of ligand molecules with known biological activity. Okay, this is the main difference between structure based drug designing and ligand based drug designing. Is that understandable so far?
Yes, sir.
Yes, right. So that is the thing, right? So moving to the original topic, right? So as I said, that the first topic in this unit is so role of X-ray crystallography in structure guided drug designing. So first part is the structure based drug designing, right? So we'll cover the importance of X-ray crystallography in solving the structure. Then we will learn the molecular docking algorithms, right? So moving on to the first topic. I hope that many of you, right, so BTech students may already studied this in some other paper like protein engineering or structural bioinformatics in detail, right? Uh, as we know that the starting point for structure-based drug designing is the 3D structure of the drug target, and whenever the 3D structure of the macromolecule is solved, either using X-ray crystallography or NMR or electron microscopy, normally the scientists used to deposit that structure in some public repositories like protein data bank. So if you go to protein data bank, we can find the solved structure of many drug targets, either only as a protein, apoprotein, or along with a co-crystallized ligand molecule, right? So if you look at the statistics of PDB, even though we have different methods for solving the 3D structure, the most predominant method is X-ray crystallography. Many structure, if you look at the percentage of structure solved for the structures deposited in protein data bank, most of the percentage will go for the X-ray crystallography because that is most well used method for solving the 3D structure, and that's why X-ray crystallography plays a very important role in structure based drug design, right? uh, okay, so is my slide fully visible for you? I think so. It's a PDF file. I hope it is clearly visible for you, right?
Yes, sir.
Yes, right. Okay. So as I said, that X-ray crystallography, it is the base for the structure-based drug design. We can say it's a backbone, or we do molecular docking, right? So we take structure from a protein data bank, and uh, we used one HSG1 or one HSG. I don't remember. HSG1, I think, right? So this is actually the structure of HIV protease, which is solved using crystallography, X-ray crystallography method, right? So my talk will be more towards the drug design perspective, right? So either we can solve the protein structure or protein ligand complex. Very important. But if you want to work with structure based drug designing, it is always recommended to have the protein ligand complex, not the protein alone. Can you tell why? So why do I say that it is always better to have the protein ligand complex rather having only the protein? So is there any reason for this? Okay, if no response, we can skip that, right? Doesn't matter. So there are certain questions that we have to address if we go for X-ray crystallography method for solving the 3D structure. So here I have mentioned the important questions that has to be addressed. So what is the validity of the particular protein structure, right? So you are taking one HSG or HSG1 from protein data bank. So what's the validity of that particular protein structure? How correct is that? Okay, first question we had addressed. The second question is how that structure is relevant to the biological acting of the protein, because you are capturing only one snapshot in the X-ray crystallography. So how that captured snapshot is relevant to the biological acting of the protein, because the proteins, they can undergo very rapid conformational changes. You might heard that some protein molecules present in its active conformation, same protein molecule can be present in different conformation called as inactive conformation. Some protein molecules you might heard that open conformation, some as closed conformation, right? So if you take any protein molecule from PDB, so how that structure is relevant to the biological acting? So how do we say that that conformation is the biologically active conformation? And how to account for the conformation of the bound ligand molecule, whether the bound ligand molecule represent the productive form or not, right? So these are basic questions that we have to answer, right? We'll address one by one. So basic concept of X-ray crystallography is crystallization. The main job of X-ray crystallographer is that they have to crystallize. Some proteins they can easily crystallize. Some proteins may be tricky for them to crystallize. It's a kind of art. More than science, it's a kind of art. So in different combination, using different solvent, adding different ions, right? So they will try to first form the crystal. So formation of crystal is the very first step, starting point. If you don't have the crystal, there's no point of talking about the crystallization. There's no point of talking about the X-ray crystallography, right? So the basic requirement for a crystal structure is the crystal. We have to form the crystal. That's the main job of the crystallographer, right? So it's a kind of art, right? So that's what I said. Crystallization of protein is still more an art than a science. It's a very important fact, right? So if you ask a crystallographer, they can accept that it's a kind of art rather than a science, right? So how much protein is needed for crystallization? If you want to crystallize a protein, how much protein is needed? That depends. Sometimes you may need very less quantity, uh, a milligram, microgram. That quantity may be enough, right? Sometimes you may need buckets of protein molecule. That depends how much similar protein is already crystallized, whether it is a very new protein or already existing protein, whether the conditions support the crystallization or not. All those decide how much quantity that we need. So even if you form crystal, right? So another important factor in X-ray crystallography is that the crystal that you have made should have the ability to diffract the X-ray, because you are going to pass the X-ray to the crystal. If the crystal is not able to diffract the X-ray, then there is no point of talking about the X-ray crystallization for that protein molecule. So if any protein does not crystallize, then we cannot proceed with X-ray crystallography. There may be many protein molecule which may not be able to crystallize by the crystallographer, right? Very important point, right? So bad versus good crystal. So here I have actually two images, right? So let me zoom slightly. Hope my slide is visible. Is it visible?
Yes, sir.
Okay. So here we have two images, right? So one is the bad crystal, another one is here is the good crystal, right? So here you can see that the right hand side, right? So good crystal, you have well defined shape. The crystal is very well formed, right? So if you diffract this, definitely, if you pass the X-ray, definitely there will be some kind of diffraction. But if you look at this, the bad crystal, why do we call it as a bad crystal? Because there's a kind of protein aggregation. You can see a lot of aggregation is there. The crystal is not well defined. It's not well formed. So if you pass the X-ray to the bad crystal, you won't get good diffraction, right? So that's how we can classify good crystal and bad crystal. So what we expect from the crystallization is that we expect always a good crystal, because the X-ray diffraction, the quality of the X-ray diffraction that depends on the quality of the crystal, right? So the basic concept of crystallization, once if you obtain the crystal, okay, so using some method, you form very good crystal, not bad one, very good crystal. So you tested the ability of that crystal to diffract the X-ray. If the diffraction takes place, then you will get the data. That's called as the data collection in X-ray crystallography. So very important definition in terms of physics related to crystallization is that related to diffraction is that diffraction pattern of an object is the Fourier transform of the object, and the inverse Fourier transform of the diffraction pattern will give the model of the object. Not much important for us, but in terms of physics, right? So this is how they get the model, right? So they first collect the diffraction pattern, and diffraction pattern is nothing but it's the Fourier transform of that object, and the inverse Fourier transform, the physicist will do that inverse Fourier transform of the diffraction pattern. They will do the calculation to get the model of the object, right? So the quality of the electron density map is going to determine the quality of the structure, right? So why do we study all this? Because we are going to take the structure from protein data bank, and we are going to use, we are not a crystallographer, right? So we are going to use somebody else deposited structure, and we should know whether we can use that structure for our study or not, whether that deposited structure is of good quality or not, right? The quality of the final model structure depends on the quality of the electron density map. Again, I repeat, for the MSc students, the quality of the final model, which is deposited in the protein data bank, that depends on the quality of the electron density map, right? So the quality of the electron density map, it depends on the quality of the diffraction pattern. Everything is interrelated. So if you get a quality diffraction pattern, then you'll be getting the quality electron density map. If the diffraction pattern is not of good quality, the electron density is not going to be of good quality. So coming to what are the factors which determine the quality of the diffraction pattern. So the first factor which determine the quality of the diffraction pattern is the intensities of the reflection. So if you pass the X-ray, right, so reflection is going to take place, how intense that reflection. So that is one important factor that is going to determine the quality of the diffraction pattern. And the second important parameter that is going to determine the quality of the diffraction pattern, in turn, that is going to determine the electron density map. Quality of the electron density map is the resolution of the data set, right? The number of criteria used to determine the quality of data set, not much important for our discussion, right? So anyway, the parameters which influences the quality of the diffraction pattern, and in turn the quality of the electron density map, or intensity, radiation damage, overlap, or merge, compactness. These are different physical parameters which is going to determine the quality of the diffraction pattern, right? So here the resolution of the data set, okay, I can add few more points, right? So resolution we define in terms of number, some number in Armstrong unit, right? So if the number is less, so this resolution is something related to your your camera lens, right? So we call it as 10 megapixels camera, right? So we call 50 megapixels camera, right? So if the number increases, the quality of the image will get increased. Just opposite to that, if the number decreases, okay, if the number decreases, say 1 angstrom, here not megapixel, angstrom, say 5 angstrom, 10 angstrom. If the number is less, then that resolution is of the best resolution or good resolution, right? So here we have example for three different resolution. So one is at 1.2 angstrom resolution, the another one is at 2 angstrom resolution, the another one is at 3 angstrom resolution. So this is a diffraction pattern. So what you are seeing here, okay, the dots circle, right? So that's a diffraction pattern at different angstrom. So at 1.2 angstrom you can find very minimum detail, right? So 2 angstrom, some details, more details there. 3 angstrom, some more details. Sorry, at 1.2 angstrom you can see more detail. At 2 angstrom, less detail, and 3 angstrom, less detail, right? So here, if we convert this diffraction pattern into electron density map, so the right hand side image, that's the electron density map, right? So 20 amino acids, right? So I hope you can remember the structure of 20 amino acid. So here, if you look at this shape, okay, at 3 angstrom, if you look at this, maybe I change the color. If you look at this shape, right, so this clearly dictates that there is possibility for some cyclic structure that we can fit, but it is not very clear. Okay, I cannot say whether it is a phenylalanine or tyrosine. Okay, or I cannot say whether it is a histidine. If you look at, anything can be fitted inside this electron density map. This 3 angstrom is not so clear to define which amino acid can be fitted inside. But if we go to 2 angstrom, right, so slightly better view, right? So we can say that definitely it's not for a 5-membered ring. Some 6-membered ring can be fitted inside, right? But we are not sure whether it's going to be a phenyl ring alone or some OH group is also there. Okay, but if you go for better resolution, 1.2 angstrom resolution, now it is very clear, right? So the atomic details there, right? So there's atom here, atom here, atom here, atom here, atom here, atom here, atom here. So if I connect all this point from this electron density map, right, so definitely I can say that, so this is a phenylalanine. Okay, so this amino acid is a phenylalanine. It is not tyrosine, or it is not a histidine or any other amino acid, right? So more details we capture if we go for higher resolution, that is less in the number, right? So less details are there if we go for higher value, okay, lower resolution, right? So 3 angstrom is no, comparatively the resolution is bad comparing to the 1.2 angstrom resolution. So is that understandable?
Yes, sir.
Okay. So what's the end point of the experiment from the X-ray crystallography? The electron density map is the end point. Okay, so the physicist will do the mathematical analysis on the electron density map. So what normally they do is they go for the shape fitting and building a model into the electron density map, right? So as I said, that resolution plays a very important role, and the measure of level of detail with which the protein view, that is based on resolution. So 1.5 angstrom or less than 1.5 angstrom is considered as the atomic resolution, which means that is a good resolution. For docking or any other in silico analysis, right? So we should not go beyond 3 angstrom resolution. So if you go beyond 3 angstrom resolution, more details will be missing, and there may be a lot of error in the structure, right? So any error in the structure will be carried out or replicated if we take that structure for any in silico work related to drug design, right? Um, so what we can observe at different resolution, because we are saying that resolution is one of the very important factor, right? So if you take the protein molecule at 5 angstrom resolution, so we can clearly see the limit of the protein molecule, where the protein is present, where the solvent is present, where's the boundary, which area is the boundary between the protein and the solvent can be very easily observed. overall shape, right? So even if you take this 3 angstrom as an example, right? So the overall shape of the atom's molecule is clearly visible, right? So at 5 angstrom also we can clearly see the overall shape of the molecule, and elements of secondary structure to some extent can be visualized, whether it is a whether that amino acid can participate in alpha helix or beta sheet. Those can be observed at the at 5 angstrom resolution. If we go for 3 angstrom resolution, so we can see general course of the polypeptide chain, shapes of side chains are very clearly interpretable if we go for the 3 angstrom resolution. But if you go for the atomic resolution 1.5 angstrom or lesser than 1.5 angstrom, 1 angstrom, right? So these are very good, right? It's very good resolution for computational studies. So individual atoms can recognizable, right? So through the electron density here, you can see if I zoom little bit, right? So at 1 angstrom, so even comparing to 2.5 angstrom, 1 angstrom is much better, because each and every atomic details are clearly captured. So anybody know what is this amino acid? Yeah, 5 membered ring attached to 6 membered.
Tryptophan, sir.
Okay, this is the tryptophan, right? So, but if we cannot see whether this electron density corresponding for tryptophan at the 4 angstrom resolution or 3 angstrom resolution, to some extent we can say that at 2.5 angstrom we can make a guess, but at 1 angstrom definitely we can say that side chain is corresponding for the tryptophan. So that is why we say that the resolution plays a very important role. So whenever you choose any protein for structure based drug designing, you have to look for the resolution. The number should be less, right? Um, so any crystallographic experiment will not provide a very accurate model. This is a very important statement for a drug designer. So if you take any structure from protein data bank, right? So you cannot trust that protein structure completely, or you cannot believe that protein structure completely. Okay. So it gives some precise model, right? So the crystallographer itself will go for a protocol called as refinement. So which means that they construct the model again and again and again repeatedly, called as iteratively. So they model iteratively and match to the electron density and fit the measure, right? So the measure of success of the fitting is given by another value called as R factor. So two important parameters. One is the resolution. Okay, resolution for MSc students, very important. The second important factor is the R factor. R factor dictates, the measure of success for that fitting procedure, right? So both the values we have to look into account, right? So this is the overall picture what we have discussed. So first, we have to make the crystallization. Then if you successfully made the crystal, we have to pass the X-ray to that crystal. So passing the X-ray, reflections will take place and will lead for the diffraction pattern. So Fourier transform and inverse Fourier transform will lead for electron density map, right? So we have to fit. Okay, so we have to fit the atoms, and you will get the model. So if you are not satisfied with this model, it's iterative process. Again, go back to the diffraction pattern and make the fitting again and again and again, unless and until you are satisfied with your atomic model. It's an iterative process, not uh, one, right? So this is very important. Initially I asked one question that as a drug designer, whenever I go to PDB, if my whatever is the target protein of my interest, I'll first look for whether I have any protein molecule which is complexed with ligand molecule or inhibitor. I won't look only for the apoprotein or protein alone, because there is no point of having the protein alone. It won't give much information for the drug designer, comparing to having the protein complexed with some ligand molecule or inhibitor, right? So for MSc students, the protein can be crystallized either as it is protein alone, or we can crystallize the protein along with some drug molecule or the inhibitor. In terms of drug discovery process, okay, the crystallographer will prefer to go for protein complexed with drug molecule or complexed with some ligand molecule. So this ligand, which includes inhibitor, okay, or drug, whatever it is, general term. Ligand is a very general term, right? So why we always prefer for protein drug complex rather crystallizing the protein alone? Because if you solve the structure of protein along with the ligand molecule, drug molecule or the inhibitor, so we can easily recognize where the binding site is present. So this is one example in which the protein molecule is complexed with the ligand molecule. The highlighting region, what I'm showing here is the ligand molecule, right? So if I have this structure, then it gives very clear idea that this is the place where the drug molecule should go and bind. Okay, that is why we always look for the co-crystallized structure, protein ligand complex, comparing to the protein alone, right? So I think you used HSG1 or 1HSG. I don't know what is the exact name, whatever it is, right? So can you tell whether it is a protein alone structure or protein complex with some inhibitor? You completed two exercises. Whether this is the structure of protein alone or protein complex with some inhibitor.
Protein complex with
It is
ligand.
protein complexed with a ligand. So this is HIV complexed with ligand molecule called as indinavir. So what you did in the first two exercise, second and third, rigid and flexible docking, is that you did a concept called as redocking. Okay. So protein complexed with the ligand. Again, I repeat, that gives information about where the binding site is present, right? Uh, okay, I skip few. Okay, this is the last slide. Okay, so uh, I hope my slide is clearly visible, right? The entire slide.
Yes, sir.
So I give a minute time for you. Can you tell both the proteins are same? Say A, B, both are same. Can you tell what differences you observe between the image A and B? Image A is no, the protein, same protein crystallized at the pH 7.5. B is again the same protein which is crystallized at at the pH 4.5. pH 7.5 and pH 4.5. Can you tell what difference you observe the structure? The protein is same. Just look at the image, compare the image, and tell me what differences you observe. Any differences you find? Anywhere you find difference? Or both are same?
No, sir, they are different.
They're different, right? So I just highlight, can you agree with me that in this portion we have some difference? So do you agree that in this portion we have some difference? Looking for some response.
Yes, sir.
Yes, right. So these are the regions, right? So but if you compare this region, I make different color, right? So maybe this green color, this region, this region, this region, these are same, right? The protein is same. Again, I repeat, the protein molecule is same. Only difference is the pH. Only difference is the pH. This is at 7.5 pH and this is at 4.5 pH, right? So at 7.5 pH, okay, the binding site of the protein molecule uh, uh, is looking one way. At 4.5 pH, the binding site is looking in a very different way. Do you agree?
Yes, sir.
Okay, now I put a question that, say for example, the crystallographer crystallized the protein at say 7.5. Okay, so 7.5 is acidic or basic or neutral? pH of 7.5, is it acidic or basic or neutral?
Sorry, above 7 base, sir.
I don't. What you said? Can you say again? Some background
Sorry,
noise.
Above 7 bases pH.
Base. That is your answer, right?
Yes, sir.
Okay, 4.5 is slightly
Acidic.
acidic, right?
Yes, sir.
Okay, so slightly acidic and slightly basic, right? So let us assume that the X-ray crystallographer, right? So crystallize the protein at the pH 7.5. Okay, the PDB structure is solved at the pH 7.5, but in reality, your protein is present at the pH 4.5. Again, I repeat. Let us assume that you don't have the structure in PDB, protein data bank. You have only one structure. That is the first structure. The first structure, the X-ray crystallographer solved at the pH 7.5, because that is the suitable pH at which he is able to crystallize the protein. But in real scenario, if your protein molecule in the body is present at the pH 4.5, okay, from this example we can say that the binding pocket amino acids, its conformation differ. But if you use this protein molecule which is solved at 7.5 for your molecular docking, for discovering the drug molecule, will you end up with a successful drug molecule? Is my question understandable?
Yes, sir.
So will you end up with successful drug molecule or not?
Not, sir.
No. The answer is going to be no. A big no, right? That is why we cannot always, take the structure from PDB, and you have to use your biological knowledge, and you have to decide uh, for the same protein molecule. There may be a lot of structure deposited in the protein data bank. If you take HIV protease, there may be hundreds of proteins which may be deposited in protein data bank. So how to choose which one is the best? You have to think of what is the resolution. Okay, which one is having the better resolution? What is the R factor, right? So which one is having the better R factor? At what pH they have crystallized the protein molecule and solved the structure? At what pH the protein molecule normally present inside the cell? So all this should be accounted. So these are considered as some of the limitations. We cannot say limitation. Any method will have some limitation, right? These are some of the limitations, which means that this X-ray crystallography cannot capture the dynamics of the protein molecule. It can capture only one snapshot of the protein structure. So you have to think as an end user whether you can use that structure for your computational studies or not. This is how the X-ray crystallography can play important role in the structure-based drug design.